Have you ever stopped to think about what's going on inside of you, right now, on a scale so small it's almost impossible to imagine? We think about cities or countries as complex, but the most incredible, bustling metropolis in the universe might just be a single one of your cells. Today, we're going to take a journey deep inside this tiny city. We're going to follow the life of one single protein from the moment it's created all the way to its final destination. So, let's dive in. Okay, first things first, let's get our bearings. Not all cells are built the same way. On the left, you've got a bacterial cell. Think of it like a super efficient one-room apartment. Simple, right? Everything it needs to do just happens in one open space. But then, on the right, you have an animal cell. And this is no apartment. It's a sprawling metropolis, just packed with specialized districts we call organelles, and each one has a very specific job to do. It's way bigger, way more organized, and navigating it is a whole different ballgame.
So, to really follow our protein's incredible journey, we first need a map of this cellular city. Let's explore the layout.
These districts of the city are the organelles. Each one is its own distinct compartment, all wrapped up in its own membrane. You've got the key players, right? The nucleus, that's the city's command center. The endoplasmic reticulum, or ER for short, that's the main factory floor. The Golgi is like the central post office. Mitochondria are the power plants, and lysosomes act as the recycling centers. What's really fascinating, though, is that the layout of this city totally changes depending on its job. A muscle cell is going to look completely different from a brain cell.
Here, take a liver cell, for example. Just look at its volume. It's totally dominated by two things: the cytosol, which is kind of the cell's internal environment, and a massive number of mitochondria, those power plants. This tells you a story right away. Liver cells are absolute power-hungry workhorses. They are constantly burning energy to make proteins, synthesize lipids, and detoxify our bodies. A skin cell or a neuron? It'd have a completely different blueprint.
I mean, this level of organization is just incredible, but it wasn't always like this. So how on earth did life evolve from that simple one-room apartment to this insanely complex, compartmentalized city?
Well, the leading theory is this fantastic story of transformation. It all starts about 1.6 billion years ago with an ancient cell called an archaeon. First, it lost its rigid outer wall. This made its membrane flexible, enough to start folding inwards on itself. Over a long time, these folds eventually wrapped around the cell's DNA, creating a protective compartment, the very first nucleus. But the final step was the real game changer. This early cell engulfed an oxygen-using bacterium, and instead of just digesting it, they formed a partnership. That bacterium became the mitochondrion, a dedicated power plant that provides all the energy needed to run this complex new city.
Okay, so now that we have the map of the city, we can finally follow our protagonist, a single protein, and its story begins with a blueprint held deep inside the command center. Our journey kicks off right here, inside the nucleus. This place is like City Hall and the Central Library all rolled into one. It contains the genome, the complete set of DNA blueprints for every single protein the cell will ever need to make. And it's all kept under the tightest security.
So, how does a blueprint actually become a protein? It's all thanks to a process called the central dogma. First, inside the nucleus, the specific DNA gene for our protein gets copied into a temporary message. We call this messenger RNA, or mRNA. That step is transcription. Then, that mRNA message gets shipped out of the nucleus into the main part of the cell. Finally, a little molecular machine called a ribosome comes along and latches onto the mRNA. It reads the message and starts stringing together amino acids in the exact right order. This is translation. Our protein is officially being born.
But just being assembled isn't enough. Not even close. Right now, our brand new protein is just a long, floppy chain of amino acids. It's not functional at all. To become a working machine, it has to immediately enter the next district, the endoplasmic reticulum, the vast industrial heartland of the cell.
And the cell is just ridiculously efficient about this. It doesn't wait around for the protein to be finished. The process is called co-translational translocation. As the ribosome is building the protein, the whole complex gets dragged to the surface of the ER, and the growing protein chain is threaded directly through a channel into the factory. So translation and translocation are happening at the exact same time. Zero delay.
Now, inside the rough ER, a supercritical quality control process begins. The protein chain gets folded into its very specific 3D shape. Think of it like cellular origami. Stabilizing bonds are formed, and a complex sugar chain gets attached, which acts like a quality control sticker. And the ER is strict. I mean, really strict. Only perfectly folded proteins are allowed to move on. And this is so important because a single misfolded protein can cause huge problems, leading to diseases from cystic fibrosis to Alzheimer's.
All right, our protein passed inspection, it's folded, it's stable, and it's ready for work. But where does it need to go? Well, its next stop is the city's central sorting and shipping facility.
Welcome to the Golgi apparatus. If the ER is the factory, then the Golgi is the post office and a finishing school, all in one. It's here that proteins and lipids arriving from the ER get modified even more, sorted, and then packaged up for delivery to their final destinations.
The Golgi isn't just one big blob. It's got a really clear direction of traffic, like a one-way street. Our protein arrives at the receiving dock, which is called the sys face, and it always faces the ER. Then it moves through the middle stacks, the medial cisternae, where it might get a few more tweaks. And finally, it reaches the shipping dock, the transface, where it's going to be dispatched.
So you might be wondering, how does our protein physically get from the ER to the Golgi and then on from there? The answer is this amazing process called vesicular transport. It uses these tiny, membrane-bound bubbles called vesicles. You can think of them as the cell's cargo containers. A little piece of the ER membrane, just full of finished proteins, literally buds off, travels through the cell, and then fuses with the Golgi, dumping all its contents inside.
So, after being processed and sorted by the Golgi, our protein is now packaged up into its final vesicle, stamped with a destination address. But where could it possibly be headed?
One possible fate for our protein is, well, it's a hazardous one, the lysosome. This is the city's recycling facility and waste disposal unit. It's filled with powerful digestive enzymes. To protect the rest of the cell, these enzymes are kept locked inside and are only activated by the lysosome's superacidic environment. Their job is to break down worn-out parts and unwanted junk. If our protein is one of these enzymes, its life's work is in this harsh, destructive place.
Another key destination is the mitochondrion, the power plant we met earlier. Now, proteins destined for this place follow a completely different path. They are fully synthesized and folded in the main part of the cell first, and then they're imported. This process is called post-translational import, which is a huge contrast to the thread-as-you-go system we saw at the ER. Or, and this is really cool, maybe our protein's job isn't inside the cell at all. If it's a hormone, like insulin, for example, its vesicle travels all the way to the outer edge of the cell, fuses with the membrane, and then releases its contents to the outside world. This elegant process of cellular export is called exocytosis.
Okay, so we've seen the whole journey, the factories, the post office, the final destinations. But this brings up a huge question. How does a protein know where to go? It turns out it all comes down to a secret language that's built right into the proteins themselves.
This language is made of something called sorting signals. These are just short stretches of amino acids, right there in the protein's own sequence, that act like a biological zip code or a shipping label. A specific sequence means, send me to the nucleus. A totally different one means, I need to stay in the ER. The entire unbelievable trafficking system of the cell is built on reading these little tags.
And get this isn't just some abstract idea. This specific sequence of 8 amino acids you see right here, that's a real-life nuclear localization signal. It acts just like a key. If a protein has this exact tag, the cell's machinery knows to transport it into the nucleus. And if you change just one of those amino acids, the key breaks, and the protein gets stranded outside. The system is that precise. It's incredible.
So who's reading these zip codes? A family of shuttle proteins that are basically molecular chaperones. You have nuclear import receptors that grab onto those nuclear import signals and escort their cargo into the nucleus. And on the flip side, you have nuclear export receptors that recognize export signals and guide their cargo out.
Okay, that makes sense, but it raises one final really crucial question. This constant shuttling back and forth can't be random, right? It needs energy, and more importantly, it needs direction. So what's powering this whole system?
The answer is just, well, it's brilliant. It's an elegant molecular switch called RAN, which uses the energy molecules GTP and GDP. Here's how it works. The cell maintains this really steep gradient. There's a ton of RAN bound to GDP out in the cytoplasm and a ton of RAN bound to GTP inside the nucleus. This difference provides direction. So when an import shuttle gets into the nucleus, all that RAN GTP binds to it and forces it to let go of its cargo.
And for an export shuttle, the opposite happens. It has to bind to RAN GTP before it can even pick up its cargo to leave. It's this beautiful system that ensures cargo is always dropped off and picked up in the right place, powering the entire import-export business.
So let's just recap for a second. A protein's entire life is this epic story. It goes from a blueprint in the DNA to being built by a ribosome, getting checked for quality in the ER, sorted in the Golgi, and then delivered to a precise address. And it's all governed by a secret language of internal zip codes and powered by this elegant molecular energy gradient. It is a logistical network that honestly makes our own global supply chains look simple.
And that really leaves us with a final, kind of mind-blowing thought. We've just explored the breathtaking, organized complexity inside one single cell. So what does it mean that trillions of these microscopic cities are working together, right now, communicating and cooperating to create the single conscious being that is, well, you?